The protocol we're going to go through next is DNA purification using a DNA cleaning concentrator kit. And this can be used when you have a solution of DNA, like from a PCR or a reaction, it could be from a digest when you're cloning, and you need to take that mixture of DNA and maybe also enzymes and other things from your reaction and purify the DNA from that solution. So in this lab, you'll use this to purify PCR products right before you make riboprobes, right before you make double-stranded RNA. You'll use this protocol to clean up a digested PJC 53.2 right before you do a ligation when you're doing cloning. But you can also use this protocol anytime you need to purify DNA from a reaction solution. This is not to be used for mini prep, so if you're growing up bacteria to purify DNA, you need to use another protocol, which is the mini prep protocol, and will be covered in a different video. So the next protocol we're going to work on together is using the DNA cleaning concentrator kit. This kit, I'm at my bench in the lab. These kits are stored above the lab computer that's close to the door of the lab, and they're made by a company called Zymo, and they look like this. You can bring this whole box over to your bench when you're ready. These kits are pretty nice because they include a protocol with them. So you can see inside the box, there's one of these. You can also look this up online and print out your own copy if you'd like. And it will tell you all of the steps that you need to do for this method. The kit will also have a few other things in it. It will have DNA binding buffer, and it will have DNA wash buffer. This is supposed to have ethanol added to it, and as long as this box is checked, where it says ethanol added, you're good to go. If you opened a new box or had a new bottle of this and this box weren't checked, you would need to follow the instructions on the bottle, and this says to add a certain volume of ethanol. Once you do that, you can check it off, and then that bottle is ready to use. The other component of the kit is a set of tubes called collection tubes. They look like this. You'll need one of these for each of your samples. And then there are also columns. And these are the Zymo Spin 1 columns that look like this. There are also columns in the mini prep kit that look a little bit different. And so if you've seen both of these, the mini prep columns have a wider filter at the bottom than the ones for the cleaning concentrate. And you're going to have one of these per sample. You're going to put them into the tube like that. So for each of your samples, you're going to have one of these, and you're also going to need one 1.5 ml tube sterile. So they're in a bag like this. You can pull out one of them and cap it. And you need to label these tubes. So if you have 10 or whatever different samples that you're going to purify at the same time, you don't want them to get mixed up. So I label the side of the column, not the collection tube, because they can get mixed up. So I just have 20 samples. I'm numbering them 1 to 20. So I'm going to label this one 1, just like that. And you're also going to label your 1.5 ml tubes. So I'm purifying PCRs, so I'm going to label them PCR and then the number, and I'm going to write CL, which is to me a note that this has been cleaned up. And then I'm also going to write the date on the lid of my tube. You can also put your initials on the side if you'd like. Okay, so I'm going to label all of my tubes and be back in a moment to move forward with this protocol. Okay, so I'm back. I have labeled my tubes and columns, and I've also gotten out my DNA that I'm going to purify. In this case, these are some hot start PCRs that I've set up, and I'm going to purify the DNA from those using this DNA cleaning concentrator kit. I've also run out these samples on a gel prior to this protocol to make sure that I have a a product for each reaction. So I've done my DNA gel to make sure I have something. You don't want to just assume that you have DNA and do this purification. It will waste reagents. So you want to run out of gel first. So again, this kit has a protocol in it. So I'm just going to follow this protocol. This says in a 1.5 ml tube, add 2 to 7 volumes of binding buffer and then transfer into the column. That's the second step. I sort of combine these and just transfer the PCR directly without going into another tube. So I'll show you how I do that. The first thing I'm going to do is I'm going to take my lids off of my PCR tubes. So they're in a little rack. And I am going to look at this little chart. And I have PCR products, but they're pretty short. So I'm actually going to use the 7:1 ratio and use 350 microliters of the binding buffer per sample. You'll notice when you try this that that's more than will actually fit in a PCR tube. So I'll kind of show you how I do it. So I'm going to pipette up the binding buffer carefully. And then I'm going to take my tubes. I'm going to try to hold this so you can see what I'm doing. I'm going to pipette this into the tube. If I pipetted the whole thing in there, it would overflow. So I'm going to just pipette up and down just once. And then I'm going to move this into my first column. Then I've left some liquid behind. I'm going to come back with the same tip and pipette that into the same column. So I'm going to do the same thing for all of my samples, where I add the DNA binding buffer to the PCR tube. I don't let it overflow, and then I just pipette it into the column. And you can, I'm holding this up, you can just leave it in a rack, but I just wanted to show you that I'm just pipetting this in there, and that will just sit on that column. And these columns are made so that the DNA will bind to the filter. It's made of a material that will bind to DNA in the presence of this DNA binding buffer. The DNA will be stuck to that filter during the binding buffer step and also the wash buffer step. And then at the end, we're going to elute the DNA, and the water will cause the DNA to come off of that filter and go into the tube. So I've done 2 of these. I'm going to hit pause while I finish them and then come back. Okay, and just to clarify with that, I've now added my binding buffer to each sample, and I have transferred them into the tubes. I wanted to clarify, I had used the 7:1 and I had 50 microliters, so that's 350 microliters. For PCR, you can also use 5:1 and it will also work. And so you may try in your own hands if you think 7:1 or 5:1 works better, but either could work. The other thing I didn't explicitly say was that I was using a new tip every time I went into the DNA binding buffer. You don't want to be pipetting DNA and then go into the binding buffer and stuff. Then it'll all get mixed up and contaminated. So you need to use a fresh tip each time you go into the binding buffer. And the last thing I wanted to say was, you may have many samples and you need to be very careful when you're pipetting that you keep them straight so that, sample 1 goes into tube 1 and so on. You don't want to mix them up. You don't want to put them in the wrong tubes, or else your downstream applications may also have mixed up samples. Okay, so when I'm done with the DNA binding buffer, I always put it away. This may seem a little silly to you, but especially when you're kind of in the middle of doing something, I kind of like only having out what I still need, so I'm less likely to go and accidentally use that binding buffer instead of the wash buffer, which is what I need. So you could only get it out when you need it and then put it back if you'd like. I think that's about it. So we've now gotten through the first step and the second step on this. The next is to centrifuge for 30 seconds and discard the flow through. And if you see up here at the beginning somewhere, I think it says perform all centrifugation at greater than or equal to 10,000 Gs. So I'm going to take you and my samples over to the centrifuge. We have 2 centrifuges in the lab. On the side nearest to the entrance of the lab, we have this centrifuge, which is a temperature controlled one. This is the one I'm going to use today. I can show you that in the other part of the lab, there's also a second one. This one can also be used. And this one is not temperature controlled. This purification needs to happen at room temperature. So if you're using the temperature controlled centrifuge, you need to make sure that the temperature is set at about room temperature, which is 24, 25 degrees or something along those lines. Okay, so centrifugation is going to pull the liquid from the it's going to pull the liquid down from the column into the collection tube. So I'm going to open the centrifuge. It says a button that says open. There's a power button here if it weren't on when you got here. You're going to open it up. There's a lid on the rotor of the centrifuge, and you can undo this rotor. Please be careful with this lid. It's if it gets dropped, it can get bent, and then we have to get a new one. So then you're going to put your samples into the centrifuge. If you haven't used a centrifuge before, you want to make sure that the samples are balanced. So that means having the same number of samples on each side. So if you just put all of these samples on one side, it wouldn't be balanced, and it could end up breaking the rotor eventually, or breaking the centrifuge. So remember, this is going to spin, and it needs to be balanced when it does so. That's true of all centrifuges. So I have 20 samples. I put 10 on one side and 10 on the other side. And if you look, they're all balanced out so that the centrifuge can work properly. This needs to click into place. You're going to close the centrifuge, and it has an automatic closure system so that it will pull itself down once it's closed. We only need to spin for half a minute. And as it said, it needed to be 10,000 Gs or less. You can go back and forth between RPMs and RCF, which is Gs. We only need to be at 10,000 or more, so I'm going to go down to 12,000 RCF, which is the same thing as 12,000 Gs. Okay, I'm going to hit start, and this is going to centrifuge my samples. So after this step, there's going to be a liquid in the bottom of the tube, and that liquid is going to contain all the materials from your solution except from the DNA, which is still going to be on that filter. And so we're going to discard the flow through, which is the material that comes down into the tube, the collection tube, and keep the column for the next step. So the centrifuge is stopping. I'm going to hit open and take the lid off. And I can show you that now the liquid is in the bottom, not in the column. So what I'm going to do is, for all of these tubes, I'm going to just take them over to the sink and dump out the liquid and then put the column back on, okay? I'm going to do this for all of the tubes. So the next part of this protocol, after discard the flow through, is to add 200 microliters of DNA wash buffer, which is our second buffer. And it says centrifuge for 30 seconds again. So I'm going to pipette 200 microliters of the DNA wash buffer into each tube. You can use just one tip if you don't touch anything, but if you go into the wash buffer and then touch your tubes, get a new tip, okay? So I'm going to do that for all of my samples, and then I'll be right back. So I have added 200 microliters of DNA wash buffer to each tube, and I've placed them back in the centrifuge. I'm going to put the lid back on, close the centrifuge, and hit start and centrifuge again. If you look at the protocol, it will also say on step 4, repeat the wash step. So after this spin, I'm actually going to just add 200 microliters more of this buffer to each tube, or to each column, without taking them out of the centrifuge. If you think you need to move them back to your rack to be able to pipette more easily, that's fine too. But I'm going to, after this spin is done, add 200 microliters more of DNA wash buffer, and then do another spin just the same as the ones I've been doing. So at the end of the second spin, my samples will have a total of 400 microliters of wash buffer in the collection tube. I'm going to take these tubes and discard the supernatant in the sink. And put the columns back on the tubes like this. I'm going to put them back in the centrifuge empty for one last spin of 1 minute at 12,000 xg. And what this will do is any remaining ethanol or wash buffer that's left on the column or in the tube is going to be down at the bottom of the collection tube, so that I don't have any of that material coming along with the column into my final 1.5 ml tubes. So I'm going to empty out all of my collection tubes and do one more spin, and then we'll resume. So that clearing spin we did where we had the empty tubes is not actually on the protocol, but you can see sometimes people will write it in. As I mentioned, that last spin is just to get all of the wash buffer ethanol off the tubes. After that step, there will be just a little bit of ethanol and wash buffer at the bottom. You don't want to dump that out, or else this will be along the side of the tube and get back on your column. So what you want to do is take this directly out of the centrifuge and either put it in your rack, or take your column out of the collection tube and place it in the 1.5 ml tube that you have labeled. And this is where we are going to elute your DNA from the filters that are in the columns into your tubes for their use. So I'm just opening all my 1.5 ml tubes. It's kind of noisy, sorry. And then I'm going to transfer all of my columns from the collection tubes into those 1.5 ml tubes. And the collection tubes can just be thrown away. All of the liquids in this protocol can just go down the sink, and the plasticware can go into the trash can, with the exception of the tips, which will go into the special tip waste. So I'm throwing out my collection tubes. And then the next step will be to elute. And as I mentioned, the DNA will stick to the filter in the column until the environment changes. So there are salts and ethanol that keep the DNA bound to the filter in the earlier steps, but at the end, when you elute, that DNA gets knocked off and into solution for it to be eluted. This protocol says use greater than or equal to 6 microliters of DNA elution buffer. And there is a little elution buffer in the kit. However, we're not going to use the elution buffer for many of our applications. We're going to just use water. And the reason for that is the elution buffer has certain things in it that are really great for DNA stability, but which are not really great for some of the applications we have for the DNA that we're interested in pursuing downstream. So for example, I'm going to use this purified DNA to make double-stranded RNA, and the EDTA and other molecules that are in this elution buffer can interfere with some of the enzymes we need to use our DNA with. So I do a little bit of an extra step before I do elution. I let my tube sit for about a minute, so we're almost to a minute. In the meanwhile, I'm going to prepare to elute. So I'm going to use water instead of elution buffer. I'm going to use my DNA to do double-stranded RNA synthesis, so I'm going to elute in depth C, or RNase-free water. So I have a tube of that. Anytime you're doing RNA work, you're going to go to use those filter tubes. And I'm going to elute in 22 microliters of depth seawater or RNase-free water for my next step. If you're using, if you're going to elute your DNA for some other purpose, there could be times that you want it in a larger volume or you want just 6 microliters. So it just needs to be over 6 microliters for this to work. This also, this protocol will tell you to add the elution buffer and then transfer your column into a tube. I think that's a recipe for disaster because you could spin and lose your DNA. So I always put my columns into the my 1.5 ml tubes, which is my microcentrifuge tube, and then elute. That way I don't get mixed up and spin in the wrong tube. So I'm going to use my filter tip and my RNase-free water. I'm going to elute 22 microliters for each sample. And I'm again using just one tip because I'm being very careful not to contaminate it, but because I don't want this RNase-free water to be contaminated with any of my samples, if you touch any of the tubes, get a new tip. And I'm going to let these sit, so you might be able to see there's just a little droplet of water right on the filter in each column, rather, and I'm going to let them sit for about 1 minute. You might need to tap them just to make sure the water gets down to the filter. If you look down in the beads of water on the side of the tube, you can tap them. I'm going to set a timer and wait for 1 minute, and then I'm going to go to the centrifuge. Okay, I'm over at the centrifuge. I'm going to put my samples in the centrifuge to elute the DNA that I have been working to purify. The only thing that's a little bit different in this step is that your tubes will have the microcentrifuge tube lid kind of hanging out, and I'll show you how I handle that in the centrifuge. The centrifuge is so powerful that these lids can break off, and it can also be kind of difficult to fit everything in there. And so what I do is put all of the lids inward facing, and that helps. If you have a lid break off a tube, that happens occasionally, and that's okay. You can just flip it back on. Usually you'll hear a big pop during centrifugation if that happens. When you're finished, you should see a little bit of liquid at the bottom of your tubes. You can take these columns out and just dispose of them, and your purified DNA is present in the bottom of these tubes, which you can cap. I'm going to do that for the rest of my samples. In the meantime, you would want to make sure that you put everything away, so all those buffers and the protocol should go back in the clean and concentrator box, and the box should go back on the shelf. And just make sure you leave the centrifuge in good shape. If you spilled anything and need to clean it up, you can work on that. You can ask for help of how to do that. And so with that, we'll finish. That finishes our DNA clean and concentrator purification. And you could use this, as I said, for many downstream applications for cloning, if you're purifying a vector, and for riboprobe and double-stranded RNA synthesis, if this is a hot start PCR that's been generated for that purpose.